Hello, and welcome back to What is a Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be starting something about the genetics and the structure of the genome itself, as well as a little bit into the RNA world, but we'll get into that in a second. So we're going to just talk about essentially genes, general genetic structure in eukaryotes, a little bit about RNA splicing, and then about genetic drift, and just some general terms about some different RNAs that we'll be discussing later on, as well as I've discussed previously. So, first of all, what in the dagnabbits is a freaking gene? And a gene is just like a nucleic acid sequence that encodes something that encodes essentially RNA that is typically encoded into protein for protein expression. So, eukaryotic genes are a little bit interesting in that they have several different regions, and the typical ones are usually the most oversimplified way is you have an enhancer, a promoter, and a gene. An enhancer is a region that is upstream of the gene, and so this gene is what encodes the proteins, and it's usually about 50 kilobases upstream. Then you have a promoter, which is right in front of the gene where the RNA polymerase binds to, and then goes and translates the gene. And then you have a cistron, which is essentially just a gene that encodes peptides. In bacteria, it's multi-cistronic versus eukaryotes, it's monocistronic, which these peptides are very much useful for different functions inside the cell. So we have an enhancer, and these enhancers actually bind to these guys called activators, usually abbreviated as AC. And the activators bind to the enhancer, and they have transcription factors, TFs, that will bind to the promoters in RNA, which will help pull RNA polymerase into the promoter and then allow for the gene to be transcribed. So how the enhancer works usually is that the activator binds to the enhancer, and then it will twist up the enhancer, and then that will actually help with the promotion of the transcription factors and binding of RNA polymerase by just kind of like all congregating in this little region right here, and then so we can actually go and translate that gene. Transcribe the gene, and then translate it into a protein later. Apologies, this type of terminology is stupid. So we have different things that are known about this. We have a pseudogene. So a pseudogene is essentially just something that looks a lot like a gene, but it's missing something. It could be missing an enhancer, it could be missing a promoter, it could be missing some small section of the gene that is almost entirely a gene, but it's missing something, so it actually does not encode a protein. And so in eukaryotes, we have RNA processing. It's a thing. Whereas in bacteria, RNA processing it really isn't a thing. And essentially, you just kind of take the RNA and you send it straight to the ribosome right off the bat. Whereas in eukaryotes, you have RNA processing. You have introns. You have these regions between regions inside this gene right here that, so essentially you get like these black regions right here, these introns that need to be excised, that need to be left and removed entirely so that it can actually encode the protein of interest. So what happens is that usually the introns are around 3.5 kilobases in size, but they can also just be up to 17 kilobases. So we need to remove these guys. These guys are huge. These guys make up such a large amount of this nucleic acid sequence. And so to do that, we use RNA splicing. But first, transcription unit. And essentially what happens is that, as I was mentioning before, we just send bacteria the proteins out and the RNA into the ribosomes in the bacteria. And usually what happens is they have transcription. You have multiple different proteins at the same, essentially multiple different genes that encode different proteins, and they all just are on the same transcript, the same RNA sequence. And so you just send it off, and then that RNA will make 5 different proteins that are all useful for the cell itself. And whereas in eukaryotes, it's more complex in that we have these introns. So it's kind of like the transcription unit has a cluster of genes, but not really, because these can be just different domains of a protein. And So what happens is that you take these transcription units and then make them into a primary transcript in eukaryotes to pre-mRNA. So before mRNA, and so essentially this primary transcript is just the raw unfiltered. It goes from here to here. It goes from the start of it to the end of it with the introns included, from the promoter to the termination site. And so because it's complicated, we need to process this RNA, and we have alternative splicing, which I mentioned before, but just a little quick review essentially is that you have different isoforms where you have proteins that are encoded by the same gene but are differently spliced. So essentially we take this little 3 guy where we have exon, 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 we can pretty much usually, we have this guy at the very front, and so then we can kind of just like cut out from here to here, and we can get this guy. Now we'll encode a different protein than this guy, or if we just remove the last one entirely, the last exon. And we can get 2 different proteins with 2 different functions or similar helping functions. It's a very complex process that just depends on random things that happen randomly. Whether or not you ate a bagel for lunch, or you ate a bagel with cream cheese and peanut butter and mayonnaise. That sounds like a nasty concoction, but that can get you this gene and make you crave even more heinous things like adding ketchup on that bagel. So these proteins just kind of happen to randomly occur differently just by alternative splicing. Now, because of alternative splicing, we also find that most of the genes in the genome and the proteins of the proteome are essentially encoded by a single copy. We have about 25 to 50% of them are a single copy. They're only a single gene that encodes that specific protein in the entire genome, whereas the rest is encoded by multiple ones in eukaryotes. And then we have, the fact is that, but what happens when we have duplicated genes, when we have the same protein encoded by 2 different genes? And usually those duplicated genes are like really close by on that haploid cell. And usually they're close by within like 5 to 50 kilobases of each other. And usually these ones, this is part of the reason for a protein family and gene family. And what actually happens is you can have one of them change as the other one changes, and you can have sequence drift. Essentially, random mutations, random nucleotides just kind of messing up and being inserted randomly or changing around, that can actually have major impact and create entirely different proteins with entirely different sequences and functions. So if we just alter it slowly and slightly, we can have essentially a DNA binding protein that helps arrange the DNA slowly sequence drift into some sort of cytosolic protein that helps protect mRNA. And stuff like that can occur just by slow evolution over time. We have divergence of function and everything. And so going on from there, we have just several different RNAs that I want to just give a quick discussion of. Our RNA, ribosomal RNA, which makes up the ribosome, along with like 50 other proteins. You've got a lot of multiple copies right there in the nucleolus of the cell that are just like right there nearby with like very different spacers, but like it's very easy and very quick for the nucleus to create ribosomal RNA just because of like the location. It's all set, just constantly going. They have snRNA, small nuclear RNA, and these guys are what help facilitate this process of alternative splicing of these pre-mRNA. They have snoRNA, which is small nucleolar RNA, which are they help guide modifications to the tRNA and rRNA because the ribosome and tRNA aren't perfect and they're just a single sequence. They have a lot of different post-transcriptional modifications, as well as the tRNA has like the addendum of the amino acid. Yeah, the amino acid added onto it, but that's not guided by the snRNA, rather just like the post-transcriptional modifications. And they have miRNA, microRNA, and these guys help regulate the stability of mRNA. As I believe I previously discussed, RNA interference, miRNA are what we essentially try and mimic in that system for RNA interference. So that's a little quick overview of like what a gene looks like and a few things about alternative splicing and RNA as well as sequence drift. So stay with me next time. We'll be talking about transposons. Thank you.